This will be a review of the interactions between the cortex and hippocampus during learning, consolidation, and retrieval, with a special emphasis on the implications for studying. We could give an alternate title: The Best Study Practices Must Align with Hippocampal Function. If we want to be successful learners, we must be paying attention. What does attention do? Attention amplifies neural activity in the sensory areas that process the learning event. The neurons are more excitable for whatever input they receive. So we've learned that in the back part of the brain, we've got visual cortex and touch cortex and auditory cortex here. And of course, these will be cortical areas processing a learning event. Well, when we are paying close attention, we are literally sending electrical signals from frontal cortex circuits back to these sensory areas so as to amplify the activity here, so that the neurons in these sensory areas are more excitable and have a more intense response to the learning event. Now notice we have our little icon for a memory trace being written in the cortex here and in the hippocampus. So when we're paying attention, we have the best chance of making the strongest memory traces in the cortex as well as the hippocampus. Notice the effect of poor attention. The memory traces are weak. Focused attention, poor attention. With poor attention, we're making very, very weak cortical memory traces and also weak hippocampal memory traces. That is not ideal. We want the strongest possible memory traces in all these areas of the brain. Now let's go ahead and sit down for our first learning episode, and we're paying close attention and we're ready to learn. So the first stage of forming a memory is the encoding stage. Here's the cerebral cortex up here. We've got 3 cortical areas. Maybe that's visual cortex, touch cortex, auditory cortex. And during the encoding stage, a population of neurons in each of these cortical areas is responding to the learning event. Likewise in the hippocampus as well, because we've learned that these cortical areas send information that converges down into the hippocampus. So encoding is when neurons are responding to the learning event. And then during the storage stage, we're going to start to form memory traces of the information, both in the cortex and in the hippocampus. Now, how are memory traces formed? We get synapse growth between some of the neurons that were activated during the learning event. Notice also that the cortical memory traces are identified as weak at this time, whereas the hippocampal memory trace is strong. Now, why is the hippocampal memory trace strong? Because hippocampal neurons show high plasticity. They can grow strong synapses with one learning episode. So here we'll have a couple of hippocampal neurons that were activated during the learning event. We have some baseline level of synapse strength before the learning, and after that learning event, we have significant growth in the strength of the synapse. Hippocampal neurons are really good at growing strong synapses in response to one learning event. And we have learned a phrase that captures this idea of plasticity: cells that fire together wire together. Now, this feature of hippocampal neurons, high plasticity, this makes the hippocampus a good learner, using a metaphor, an educational metaphor. So, understanding that the hippocampus has high plasticity gives us a way to think about study techniques. How do we want to study? We want to use study techniques that are going to make the strongest possible hippocampal memory traces. Those techniques are the deep processing tasks. Deep processing simply refers to a technique that produces a memory advantage, and here we're starting to unpack what that means. Deep processing tasks tend to activate more widespread areas of the brain and do so more intensely than shallow processing tasks. And as a consequence, we'll have stronger memory traces in the hippocampus when we're using deep processing study techniques. We saw in class a couple of experiments that demonstrated the difference between a deep and a shallow processing task. So evaluating the pleasantness or unpleasantness of a word produced a memory advantage compared to counting syllables. Likewise, drawing the word produced a memory advantage compared to simply writing the word repeatedly. So these are deep processing tasks, and the idea is that they are activating more areas of the brain and more intensely, and so that then when it comes time to retrieve the words 5 or 10 minutes later, the stronger hippocampal memory traces under the pleasant, unpleasant condition or the drawing condition, those stronger hippocampal memory traces were more successful in reactivating the cortical memory traces for those words in the deep processing condition. So deep processing tasks are setting up stronger hippocampal memory traces that can be used to retrieve those weak cortical memory traces soon after learning, like after a 5 or 10-minute break. Now, what are some of these deep processing tasks? Things like concept maps, drawing information, reorganizing information. Don't just reread your notes, but reorganize it in some kind of way. It requires some thinking. Retrieval practice is deep processing. You can use the blank paper technique. You just do a data dump. You can answer questions or use flashcards slowly to learn vocab. Teaching others is very deep processing. It takes a lot of brain activity to be able to successfully teach somebody what you know. In general, these deep processing tasks require more thinking. And when we're doing more thinking, we're more likely to be setting up stronger memory traces in the hippocampus. Now, because we have these stronger memory traces in the hippocampus with deep processing tasks, we're going to have more effective consolidation when we go to sleep. We previously learned that memory consolidation occurs during sleep, and generally speaking, it's when we're going to reactivate hippocampal memory traces to stimulate the cortical memory traces to grow stronger synapses among those memory trace neurons, and we call that hippocampal replay. This diagram is a little bit different than previous ones. Instead of showing a paper with a pen applying ink for a memory trace, we're showing individual neurons here, and we've indicated with a sort of a dashed line here that the cortical memory traces were initially weak. We initially made a strong hippocampal memory trace with the solid lines here, but still the idea is the same. We're going to reactivate the hippocampal memory traces to strengthen the cortical memory traces. And the effect in the cortex then is that those cortical neurons will grow stronger synapses, and that's long-lasting growth because the cortex is the hard drive. So what memory consolidation is doing is sort of writing data to the hard drive. The hippocampal memory traces are being used to store long-term memories in the cerebral cortex. Okay, so during sleep then, it's as if the hippocampal learner becomes the teacher of the cortex. So again, the hippocampus was a good learner because it has high plasticity neurons capable of making strong memory traces for just one learning event. But then at night, those memory traces are in a sense sort of being used to teach the cortex to strengthen the cortical memory traces. Notice also that the flow of information is reversed. So at night, the hippocampus is communicating with the cortex, but when we were learning, it was the cortex that was communicating with the hippocampus. So we have a reverse flow of information, learning versus consolidation. So now, after we have consolidated memories in the cortex, we've strengthened those cortical memory traces, an interesting thing appears to happen, that the hippocampal synapses then get weakened. So the hippocampal memory traces will be weakened in the next phase of sleep. And this does serve a purpose to refresh the hippocampus for new learning. Let's take a look at that in more detail. Up here at the top here, before learning, we've got a set of hippocampal neurons, and notice the synapses are at some weak baseline strength. And then we go through a day of learning. Well, some set of hippocampal synapses here will be strengthened, and that's what represents the learned information. But there are a limited number of hippocampal neurons, limited number of synapses, and once we grow these synapses nice and strong after a learning event, these synapses cannot be strengthened anymore for further learning. We can call that synaptic saturation. So throughout the day's learning, we're going to be kind of strengthening lots and lots of hippocampal synapses, but that leaves the hippocampus unable to learn more. How do we get around that? We go to sleep. So these strengthened synapses must be reset to a baseline strength. And notice we've reset them. We've weakened them again. Now these synapses are ready to learn new information the next day. So sleep is performing two really important functions. In phase one, we're going to use strong hippocampal memory traces during consolidation to strengthen the weaker cortical memory traces, but only once that's done, then we're going to weaken those hippocampal memory traces to refresh the hippocampus in preparation for new learning. You see, the cortex is the long-term system. We want to strengthen those memory traces in the cortex, but keep our hippocampus fresh and ready for new learning. So after one learning session and a night of sleep, we still might actually forget what we studied the night before. Now, why would that be? Well, because generally we still have weak memory traces in the cortex, and we've got our refreshed hippocampal memory trace. And so if we only study once, we're in danger of not having sufficiently strong cortical memory traces that we can reliably retrieve that information. And this is one way we forget. We call that storage failure or insufficient consolidation. We just didn't study enough. We didn't study repeatedly to make these cortical memory traces strong. So if we want to be educated, we're not done yet. We've got to study a second time and a third time. And now, why is that? We can explain that by thinking about the cerebral cortex neurons. Cortical neurons show less plasticity compared to hippocampal neurons. They grow strong synapses more slowly after multiple learning episodes. So notice here we have a couple of cortical neurons involved in the learning. There's the baseline synapse strength. And in the first study session, we increase the strength to this. And then the second study session, the synapses grow a little stronger. Well, that's all well and good. But notice it takes multiple study sessions, multiple activations to grow these synapses stronger and stronger. So we can think of that as slower growth. By contrast, we said the hippocampal neurons, there's fast growth. They're very plastic, high plasticity, low plasticity for cortical neurons. And this has obvious implications for studying. We've got to repeat our study sessions. We have to repeatedly activate those same cortical neurons to really grow those synapses strong. But if we do that, if we put in the work, we're going to see the benefits of repetition. If we can drive those cortical synapses to really high strength, we're going to have slow decay of those synapses. So whatever information we're storing in our long-term memory system, we'll keep that knowledge handy because we've made strong synapses between those memory trace neurons. By contrast, you'll recall the hippocampal neurons, the synapses are weakened during sleep, so we could say that the memory traces show fast decay. But that is a design feature. That's not a bug. That's a feature. We want the hippocampal synapses to be weakened in preparation for new learning. But in the cortex, we've got to have multiple study sessions to strengthen those synapses. And once we do, we will enjoy strong memory traces because there's thought to be slow decay. Otherwise, we wouldn't have a long-term memory system. Let's use this brain diagram to illustrate the effects of multiple study sessions separated by sleep. At these cortical locations, we see 3 files here. That's our icon for a memory trace. And you'll notice as we go from the left one to the right one, there's more ink. So the memory trace is going from a weak memory trace to a strong one. Well, how do we get to the strong one? We study repeatedly, spaced out in time with sleep in between, because each time we go to sleep, the hippocampal memory traces can be used to strengthen the very same cortical memory traces. These memory traces are the ones that are storing the information for the material that we're studying. If we study the same material on multiple occasions with sleep in between, we give the brain the opportunity to do multiple rounds of memory consolidation during sleep, and that will progressively strengthen the cortical memory traces. Using the analogy, spacing out our study sessions allows the hippocampal printer to apply more ink. Now, notice we're suggesting that each time we study on successive days, we're setting up new hippocampal memory traces. So we're studying the same information, so the cortical memory traces will be the same ones that we're activating each time we study. But if we study on successive days, it'll be a different hippocampal memory trace, each of which will be used at night to do memory consolidation during sleep. Now let's examine this in a little bit more detail, what's really going on in the hippocampus. After that first study session, we're left with some consolidated memory traces, although they still might be relatively weak. So we want to study again. Our hippocampus is nicely refreshed. We've got a weakened memory trace here, and we're ready to study again, and preferably we want to do that after we sleep. So the next day, we want to study the same information again. And when we do that, it's going to be a different set of neurons will be recruited to form the memory trace during the second study session. So here's our cortical memory traces. It's the same ones that were activated during the first study session. We're going to reactivate them. It's the same information, but notice down in the hippocampus, it's going to be a different hippocampal memory trace compared to the original memory trace. And that's good. So these hippocampal neurons are fresh and real responsive, and they are capable of strengthening their synapses to make a good, strong hippocampal memory trace. So those fresh neurons have synapses that are capable of new growth to represent the new learning of the same information. Remember, it's the same cortical memory traces being activated here. Now, the new hippocampal memory trace then will be again replayed during sleep to consolidate the same cortical memory traces. So again, after this second study session, we're going to go to sleep, and now it's this new hippocampal memory trace that is linked to the same cortical memory traces that will be used to be reactivated to replay that memory trace and then to strengthen even more the cortical memory traces. Now let's also at this point add a cautionary note. Given our study of the hippocampus so far, we don't want to be using long study sessions. Continuing a long study session activates the same hippocampal memory traces. So here we have a prolonged study session, and as we just lengthen that study session to 1 hour, to 2 hours, to 3 hours, we're still just activating the same hippocampal neurons, and that is not good because continuing a long study session leads to synapse saturation and little gain in learning. Remember, these are high plasticity neurons, and they grow really strong synapses early in the learning session. So when we add another hour and then another hour after that, we're not doing ourselves much good. The hippocampus has already made strong memory traces. Further study produces little gain. Now, in a pinch, if we just have 1 day to study before an assessment, we still want to make study sessions short and then have delays between the study sessions. By creating a delay between study sessions, it is more likely that a different set of hippocampal neurons will be recruited for the second study session. Those fresher neurons have synapses that are more capable of new growth to represent the learning in the second study session. So again, let's say that these 3 neurons here were the ones activated by the first study session, and then we go take a break. The break makes it so that when we come back for the second study session, we're more likely to be activating a different set of hippocampal neurons, which are more likely to have weaker synapses, right, compared to the ones that we were just activating in the first study session. So these are fresher hippocampal neurons capable of more synapse growth to represent the learning of the second study session. The way we could look at it is that if we just keep studying for a longer and longer amount of time, study the same stuff, just more and more time, then we're just going to be saturating those hippocampal neuron synapses there. What we want to do is take a break to make it more likely that when we come back for that second study session, we're activating a different set of hippocampal neurons with fresher synapses, sort of weaker synapses that can be strengthened again for that learning. So if we use multiple and short study sessions separated by sleep or by breaks, if we're studying the day before the assessment, we get the benefits of multiple rounds of consolidation to build stronger and stronger memory traces in the cortex. And as a result, retrieval will be more successful. Notice we've got strong memory traces in the cortex here. We do have a refreshed hippocampus here, but if we make strong cortical memory traces, we'll have a good chance of being able to reactivate those memory traces and retrieve the information we want. By definition, cramming involves a single learning episode and therefore deprives your brain of the opportunity to make stronger memory traces in the cortex. Cramming gets you these synapses and weak cortical memory traces when you could have these strong synapses with repeated study. You'll be left with weak cortical memory traces and forget the information quickly. Now let's say a word about retrieval practice as an excellent study technique. We have learned one form of forgetting is loss of access, and we showed that in a diagram like this. On the left, here we have somebody who has set up strong cortical memory traces, and they have strong retrieval pathways. So they've been accessing this information on a regular basis. So this person knows what they're talking about. They can quickly and readily and reliably retrieve the memory for the information stored in these memory traces. But if they go for a long period of time where they do not retrieve this information, we'll call that disuse. What can happen is that the search systems, the memory retrieval systems in the frontal cortex, those systems can lose access to the memory traces. They're still there and they're still strong, but it's as if the brain has trouble finding those memory traces because of this long period of time where we did not retrieve the information. So retrieval practice addresses this problem directly. Retrieval practice strengthens the retrieval pathways and so prevents an important form of forgetting. You see, if we regularly retrieve information, we're going to keep the cortical memory traces nice and strong, and they don't decay very quickly at all, so they'll be strong, but we'll also keep the retrieval pathways strong. And that's really the ticket to having fast, reliable access to information. Another important benefit of retrieval practice is that it also builds stronger cortical memory traces. Now, how does it do that? Well, in this diagram, we saw what it is to retrieve a cortical memory trace. We reactivate the memory traces and load them up into the working memory system. But we also learned that anytime we activate these cortical regions, not only are they sharing information with working memory, but they're also sharing that information with the hippocampus. So a retrieved memory makes a new hippocampal memory trace for that retrieved memory. So in a sense then, retrieved memories are like another learning event. And so because retrieved memories are like a learning event, well, they too made a hippocampal memory trace that can be used during sleep when we go through memory consolidation. So a retrieved memory becomes another opportunity to consolidate the cortical memory traces for that information. So in this way then, retrieval practice strengthens cortical memory traces. Every time we retrieve a memory, we're giving the brain another chance to go through memory consolidation during sleep. In addition, we're strengthening the retrieval pathways. And a third benefit of retrieval practice we've learned about earlier is that it helps us improve our metacognition. It can show us what we know and what we don't know. So let's end this lesson series here by comparing sort of the hippocampal memory system and the cortical memory system, and notice some similarities and differences. In the hippocampal system, it's a small region of the brain, so approximately 50 million neurons maybe in the human hippocampus, whereas in the cortex we've got something like 16 billion neurons. So there's just a whole lot more neurons in the cortex than there are in the hippocampus. Let's take a look at these two memory systems. So in the hippocampus, we've got fast learning, high plasticity neurons, but fast decay. During sleep, we're going to reset those synapses for new learning. By contrast, in the cortex, we've got slow learning, right? Lower plasticity, so we need multiple study sessions to make strong synapses. But once we make them strong, we've got slow decay. And that's beneficial, right? So slow decay, that's our long-term memory system. Fast decay, that's preparation for new learning. This kind of makes sense, these two memory systems. So here we have our hippocampus, the fast learning but small capacity system, and the cortex, a slow learning but large capacity system. And then what is consolidation doing? It's integrating the day's learning into the long-term memory system. So we make good, strong hippocampal memory traces quickly of the day's learning, and then we use them at night to strengthen the cortical memory traces. And remember, in the cortex, there's a slow decay of those memory traces. Well, that's a feature of a long-term memory system. We don't want these memories to simply decay over time. We want them to be nice and strong and to keep them strong. Whereas in the hippocampus, we need to weaken these memory traces. We need to erase them so that the hippocampus is ready for new learning. And the reason for that is there are just far fewer neurons here. There's a capacity limitation here, so we've got to keep the hippocampus fresh and ready to learn. So just as it is important to know how to study, it's equally important to know when to study. We want to study when the hippocampal teacher is prepared to learn. And this means reduced synapse strength between hippocampal neurons, which occurs after sleep. Or study again when fresh hippocampal neurons capable of synapse growth can be recruited to the memory trace. This occurs after a delay since the last study session. So to conclude then, we have seen that the dynamic interaction between the hippocampus and the cortex, the way that these brain systems operate, sheds light on the best practices we want to use in our studies. We want to use deep processing techniques. They have the best chance of making strong hippocampal memory traces. We want to repeat our study sessions because these cortical neurons have low plasticity. They have to be activated many times to grow strong synapses. We want to use short study sessions because the high plasticity hippocampal neurons will reach synapse saturation very early in our study session. So we want to shorten up our study session and take breaks so that new fresh hippocampal neurons can be used for that next study session. We want to get good sleep. We want to space out our study sessions so that sleep is in between them so that we get the benefits of memory consolidation, and that occurs during sleep. That is a really important way that we are strengthening those cortical memory traces. And we want to use heavy doses of retrieval practice because that'll keep our retrieval pathways nice and strong. And every time we retrieve a memory, we get another chance to do consolidation to strengthen the very same cortical memory traces for that information we're retrieving.